Well, I just want to start off. There are going to be three topics in this lecture. First is basically what an ontology is. Then I'm going to show you two projects that are currently unpublished but will appear shortly. So why would we want to have a phenotype ontology? Well, let's just think about bioinformatics in general. And just to make things kind of simple, I mean, essentially a lot of algorithms will take multiple biological things and will calculate a similarity between them in the form of a number, and we'll use this number to rank the objects or cluster them in some way. And we're seeing here that the first bioinformatician, probably Margaret Dayhoff, and work that she did eventually led to things like the Blossom matrix, and this is used basically to rank homologous sequences. And so the question is, in medicine, how do we calculate the similarity between a patient with a cold, a broken arm, a heart attack, or sepsis? Well, I mean, it's not immediately obvious, and so in any case, the human phenotype ontology has been developed as a way of addressing that challenge. So, introduction. There are a couple of things about ontologies that I'd like to tell you today. A couple of them are completely boring, but important. And so, when we started, one of the motivations for developing the HPO was the fact that we wanted to use data in Phenome for bioinformatics research and online Mendelian inheritance in man. If you don't know it, it's one of the great monuments, I think, of research in the century. It's over 50 years old, and it's the central resource that I think many of us in genetics use. But it's not really designed for computation, it's designed for humans. And so, for instance, here, generalized myotrophy, so wasting, non-localized wasting of muscles, has at least 10 synonyms in Omen. And so if you're trying to search or do an analysis, and you want to find all of the diseases that have this feature, well, it's pretty hard. And so one of the things that ontologies do is to provide a controlled vocabulary and say that these are all synonyms so that the computer knows this. Another thing that is maddening in biomedical research are things like this: myopathic electromyography. So what does that actually mean? Is that it's actually not a phenotype? That's a sort of an odd description that evokes many things, perhaps, in your mind. But it's really not something that a computer can deal with. And if you're doing text mining and you're trying to figure out still walking 25 years after onset, I'm sure everybody here knows, well, this must be some neurological disease that somehow is very chronic, but a computer would never do that. And these are some reasons why databases today still are having a lot of difficulty to talk with one another about phenotypes, to be interoperable. And so just to start, I'll admit, I'm a nerd, so I don't like sports. I hate sports. I was happy that Germany won, but I was glad that I didn't actually have to watch the game. And, anyways, but these are two footballs that, for the computer, we have the same string of zeros and ones. So if you figure out how to code football in ASCII, that's how in bits, that's how it will look. And sort of the same thing in medicine. And just imagine that you're hopefully a long time from now on an intensive care unit, and there's this wonderful EHR that's much better than anything we have today, and this particular string of zeros and ones goes by. That means they both spell fibrillation, but if you have this, then your muscle is twitching. If you have this, well, you better hope that there's a cardiologist right next to you, because you're going to die. And hopefully your EHR will be using ontologies and know the difference between the two. And so what is an ontology? Well, do you know who wrote the first ontology? It's actually Aristotle, well over 2,000 years ago. And I'll tell you a bit what an ontology is, but he in some of his works described hierarchical systems, for instance, of animals, and also some very philosophical constructs that I don't really understand, but these were ontologies. He didn't use the word ontology. It's a Greek word. It means the study of being. But actually, in the 70s, when computer scientists in artificial intelligence were thinking about knowledge representation, so how do I, not just numbers, it's knowledge, how do I represent that in computers? Started to use this word. And actually, this turned into a hot topic in science in 1999 when 3 groups formed gene ontology. So these were people from the mouse world, from the yeast world, and I think from the rat world. And they all had databases about gene functions, but they were using different words and different systems, and they couldn't talk to one another. And so they got together and said, let's unify our systems, and that's this paper, Gene Ontology, Unification of Biology, that's been extremely influential and has spawned now hundreds of biological ontologies. So let's just ask ourselves, what is an ontology? And so I think this is kind of helpful, and this is one of, I think, the most brilliant definition I've ever seen in my life. An ontology is a specification of a conceptualization. Or the first time I saw this, I said, huh? It's not obvious, but let's go through this. So conceptualization, let's just imagine we're talking about wine. So something I like a lot. And let's imagine we have a catalog of wines. All right? And so this will be, we'll have Chardonnay, we'll have Rioja, we'll have Riesling, all of these things. And if I then have a definition, I'll have a glossary. So Rioja is a Spanish red wine. If I also have a list of synonyms, so I'm not that good at wine to know all of the synonyms, but imagine that I list the synonyms and I have a thesaurus. All of this is not yet an ontology. But this is a conceptualization because I've written down the concepts of a domain. And here we start to get into ontologies. And if I say Rioja is a particular type of red wine, then I've specified the relationship of concepts to one another. And so I can say, Campo Viejo, that's the wine we drink at home, is a type of Rioja. Rioja is a type of Spanish wine, and so on. So I have subclasses. This is now an ontology. And actually, if you look at the full semantic web, there are about 30 different ways of specifying relationships between concepts. I'm not going to talk about all of them, but just as an example, a nucleus is not a subclass of a cell, it's part of a cell. And so that's another one. You could say fibrillin 1 is not, let's say, a subclass of gene, it's an instance of gene. So it's a particular instance, let's say, and there's lots of other ways of doing this. And so why is this interesting? Is anybody a Trekkie? A few people? Well, you may recognize that this is Worf from The Next Generation. Anyway, but this is actually first order logic, but ontologies use description logic, which is almost the same thing. It looks a little bit different maybe, but what does this mean? This means for all x, the fact that x is a Klingon implies that x is an opera lover. It's not Puccini or something, there's Klingon opera, but this is actually true. And Trekkies know this to be true. This guy is called Worf, and this statement says Worf is a Klingon. And so using ontology reasoning, we can automatically infer that Worf is an opera lover. Okay, well, this looks kind of obvious and trivial, but we start off with our database, it has 2 items, and we run our program, and now it has 3 items. So we learn something new based on some rules and the data we have. And so if we start off with a database with 100,000 items, and we get 20,000 new true things, well, that can be pretty powerful. Okay, so what is a phenotype ontology? Well, phenotype in biology means things you observe. In medicine, it almost always means things that you observe and they're abnormal. And that's how we're using this word. So it could be a sign, so something a physician observes or hears with a stethoscope. A symptom is something a patient complains about. Lab findings, so blood tests, enzyme activities, imaging studies, so chest X-ray, MRI, neurophysiology, behavioral abnormalities, basically anything that a physician would use in the differential diagnostic process to try to figure out what a patient has is within the scope of the HPO. A warning, some people use the phenotype to mean disease, so they'll say the phenotype of common cold. We don't use that word in this way. We think that a disease is a disease and it has phenotypic features. So the common cold would have the phenotypic features of fever, runny nose, cough, etc. And actually, we were talking about this a couple of times today, but when I learned medicine at UPenn, which was wonderful, actually the way they taught you medicine was they showed you some slides, they started talking about patients, and you were just expected to somehow learn this by osmosis. It actually worked, but you were never really taught the precise criteria. And computers being significantly stronger than humans still, they just can't deal with this. And so essentially, there's two ways of making a diagnosis in medicine. One is somebody comes through the door and you say, oh, that looks like Down syndrome. Well, that actually does work a lot. But if you can't make the diagnosis, then you're just completely stumped. And so in medical genetics, what people learn to do is to break down what you see into individual phenotypic abnormalities. And so actually breaking down the phenotypic features of the patient into components, using a controlled vocabulary to enter that into a computer, is what's become known as deep phenotyping. And we published an issue in Human Mutation on this a few years ago that I think has a lot of really great authors, if you want to learn more about that. Anyway, so actually as a tool for deep phenotyping, we developed the Human Phenotype Ontology. And each of the terms, so I was speaking about Rioja, Spanish red wine, wine, and so on. Each of these are also in a subclass hierarchy. So atrial septal defect is a particular phenotypic abnormality, but it's also a subclass of abnormality of the atrial septum, which in turn is a subclass of abnormality of the cardiac atria, which is a subclass of cardiac malformation, and so on. Altogether, we have about 10,500 terms like this in all organ systems, and the word annotation, so for 7,000 diseases, so if one of these diseases was common cold, and my database said common cold is characterized by HPO term for fever, that's one annotation. A second annotation would be common cold is characterized by cough, and so on. And so there's 110,000 of these things for these diseases. The HPO is completely free for academics and for businesses. If you want to, you can download it and try to sell it. I'd appreciate it if I got a beer, though. So how do we use this now to do interesting work? The concept of information content is important, and so what this means is that I've got a particular term in an ontology, and this is the probability or actually frequency of that term. So if we imagine that this logarithm is a decadic one, so a base 10 logarithm, and 1 out of 10 items in my database have this term, so 1 out of 10 diseases have fever, then the logarithm of 1/10 is -1, so we have a minus here, so the information content is 1. If 1 out of 10,000 diseases have it, then the logarithm is -4, the minus here, so the information content is 4. And this is, if any of you know about Shannon's information theory or entropy, if you take the expected value of this across your database, you actually would get entropy, and there are theoretical reasons that this is a really good way to measure information. And okay, so how do we use that to work? Well, let's take a look at this mini ontology. I'll just run you through this. So this is, let's say, a database of carnivores, I've got 1,000 items in my database, and these are, I believe, these are specific items. So here's Beagle, Terrier, Husky, and so the frequency, the probability of Beagle is 40 divided by 1,000, which corresponds to this information content. One thing is that these 40 annotations get inherited up. So for dog, it inherits these annotations 40 plus 5 plus 15 or 60, and there are, let's say, another 290 annotations to dog. But because this is inheriting annotations, this is always less specific than the children, so the information content always gets less and less the higher up we go. That's important. Anyway, how similar are cheetah and lion for an ontologist? Well, we go to the most specific common ancestor, which is big cat, and we take its information content. So there are some other ways of doing it, but this is the easiest way. And if I want to know how similar are beagle and wildcat, well, the most specific common ancestor is already at the root. And since this has inherited all of the annotations of everything, its frequency is 1, and the logarithm of 1 is 0, so it's got no information content. So these are not at all similar. And for comparing genes with gene ontology, and we want to know how similar are two genes, so gene 1, gene 2, this will be annotated to two terms, C and F, and this one to F and G. And what this equation here means is for each of the terms in gene 1, find the term in gene 2 that has the maximum information content, and take the sum of all such terms. So I do this once for C and once for F. And for C, basically the most specific ancestor, I'll look here at G, F and G, and I find that that's actually B. So that's here. And for F, again, I start here, and this also has F, so I can stay there. And so I get these two terms, and this gives me this number. And it's the same for diseases. So a disease will have, a couple of phenotypic features. This one will have a couple of phenotypic features. So now I can actually get this number to measure the similarity of two diseases. And it turns out that the formula I showed you is not symmetrical, depending on the order. So we actually take the average in both directions. That's not that important. But here we took 1,000 diseases, and we took them from a database that had divided the diseases into 20 categories, such as cancer, cardiovascular, dermatology, and so on. And without looking at these labels, we clustered, we basically measured the similarity of each disease to the other. We had a matrix of 1,000 by 1,000 similarities, and every time the similarity was higher than a threshold, we drew a line between the nodes and edge and connected them, and then we filled in the color. And so basically, just based on the phenotype, we could recapitulate these classes to a great extent. And there were also things like neuromuscular. Well, for a physician, that sounds like it's right. It might even be the same clinic. Hematology, immunology is often the same. Anyway, so this gave us confidence that this formula was actually a reasonably good way of measuring the similarity between two different diseases. And we then said, so when I did my residency in pediatrics in Berlin, I wanted to do more research, so I went to the genetics department, and I was soon confronted by these 7,000 rare diseases, and it was really difficult for me because my brain doesn't do this gestalt as well as some of the experts in our department. So what you do, you write down the features you see, and at that time, you went to the London Histology database. Maybe some of the geneticists among you are familiar with that. And you would then basically say, show me all diseases that have 2 or 3 of these features. And you would get an unsorted list of 200 diseases. To be honest, I was overwhelmed. And then you would say, okay, well, let's try 3 of 3 features. And you would get no diseases. And so this didn't help me. And so we wanted to develop something that would weight the matches according to the specificity of the features and give us a ranked list. Okay, and this is what we did. Well, actually, it's the same formula that I just showed you, but we're not comparing 2 diseases. We're comparing a set of query terms. So the doctor wrote down some phenotypes, and this then gets compared to all of the 7,000 diseases in our database. And in this example, we're looking at Noonan syndrome and Opitz syndrome, or one of the Opitz syndromes. And the doctor said, well, the patient has down-slanting palpebral fissures and hypertelorism. And so our program looks in the database and says, well, Noonan syndrome has down-slanting palpebral fissures and hypertelorism. Okay, good. So I get the information content of these two terms. But for Opitz syndrome, this has hypertelorism, but it doesn't have down-slanting palpebral fissures. And so we go up to the neck, the most common ancestor, and I think that's abnormality of the eyelid, and that has a lesser information content. So our program ranks Noonan syndrome above Opitz syndrome. It does this then for all 7,000 diseases, and this is our implementation of the algorithm. It's called the Phenomizer. You can get this on the web for free. And so you would enter some terms using either a search window where you enter a few letters, or you can search within the ontology structure. I'm not going to talk about this today, but we developed a statistical method that actually improves upon the results of just the raw semantic similarity score, because it turns out that the maximum similarity you can get for some diseases is different, because some diseases are annotated to 10 terms, some to 100 terms, and so on. But that's not important for what I'm going to talk about today, but you can read about it.
That assumes, Peter, that your initial annotation is correct.
Yeah. So that's a good point. One of the things I learned at this medical school, well, there were two things. One was Occam's razor, which we all know. The other one was Hickman's dictum. Do you know that? So that is, a patient is entitled to as many diseases as he or she damn well pleases. And so, what if you enter 5 terms and only 3 match to the genetic disease, and then the patient has 2 other things? The algorithm is actually robust to that, and we also have another algorithm that basically embeds the HPO into a Bayesian network such that it downweights false positives and downweights false negatives, given that enough terms sort of match, and that does affect the performance a bit. But it doesn't, to be honest, it doesn't seem to be a very critical thing to this algorithm, because if you have 3 really specific matches, that gives you a high score, and then the others will maybe dilute this a bit, but it still seems to work in practice reasonably. But it doesn't always get you to, I'm not claiming that this will replace a physician, it's just intended to be an aid. Okay, so that I basically have shown you now some semantic reasoning algorithm, the HPO, and graph algorithm, so how we're actually measuring similarity with the HPO. I now want to show you 2 things that, one is for research, and the other is for clinical diagnostics. And this slide was sort of for bioinformatics people. It's intended to say what is translational research, but just very briefly, one of the goals is to take all of this stuff from basic science, from animal models, from numerics, and translate this into clinical care, diagnostics. So I think we've done something like this in the second project I'll show you, but also to take this data and use it to understand this. And so that's what I'm going to be talking about in the next 10 minutes. So I think this audience is a little heterogeneous, so I'm going to explain some basic concepts. Please forgive me if this is old hat to you, but we're interested now in looking at the relationship between gene regulation, phenotypes, and copy number variants. I'm just going to say CNVs. And the important bit about gene regulation for this talk is that here we have a gene that gets transcribed here. Here we have, let's say, 50,000 nucleotides, and here we have a tissue-specific enhancer. And the way this works is that you get a looping, and this the enhancer is bound by, let's say, a transcription factor that in turn binds some other proteins and activates transcription of the gene in a tissue-specific manner. Okay. This is a CNV copy number variant. So essentially, this bit here is deleted. So normally you have 2 copies, maternal, paternal. If one of them is deleted, you only have 1 copy, and so you have a haploinsufficiency of all of these genes. You have 50% of the normal gene dosage, And it turns out that some, but not all of the genes are sensitive to that. And for instance, haploinsufficiency, or if you have a mutation in PBX1 in this gene, you'll get heart defects. And this is thought actually to be one of the causes of heart defects in this CNV disorder. And some of the other genes might be contributing to some of the other phenotypic features. And the scientific question is, well, what is the relationship between these individual genes and the CNV phenotypes? The practical question is, basically, is this particular CNV the cause of my patient's problems? And today, basically, most people have been evaluating CNVs using this gene dosage theory. And so we had a couple of observations that called that into question. A lot of the next few slides are the work of my colleague Malte Spielmann at our institute in Berlin. And he was looking at Liebenberg syndrome. And so this is a normal elbow, and this is the elbow of an individual with Liebenberg syndrome. And you can sort of see that it's starting to look a little bit like a knee. And this here is the wrist of a patient with Liebenberg syndrome. So it's starting to look a little bit like an ankle or a heel there. And so there's a homeotic arm-to-leg transformation. And this was sort of a mystery for a long time. Where is this coming from? And Malte found 2 deletions in patients, in families with Liebenberg syndrome. And so he looked at this and he said, H2AFY, what's the connection between this gene and Liebenberg syndrome? He couldn't find anything. And then he looked to this side and he said, oh, Phenix1. So this is actually a Hox gene. It's responsible for hind limb patterning. And said, well, maybe that has something to do with this. And he looked on the other side, and he saw this enhancer. So if you look at the VISTA enhancer database for this, which is right over here, you'll see what's going on in the hind limbs and the forelimbs. And so the obvious theory was that this enhancer is inappropriately activating a hind limb gene, which is making the arms look like legs. Okay? And we then saw this paper from I think Stanford, the Wren group, really brilliant paper, where they were using a method called Hi-C, which measures chromatin interactions across the genome. And you may have seen this, but the way to interpret this is that if you have a red dot here, it means that there's an interaction between this bit here and this bit here. So this might be an enhancer, this might be a promoter, for instance. It's probably more complicated than that. But what's striking is that there are domains. So there's really, this bit here doesn't really interact to the right. It just interacts to the stuff in this domain. And so we were fascinated by this, and we started to re-examine the Liebenberg data. And the idea is this, that you have these domains, and here's a boundary here, and normally the boundary would prevent this enhancer from activating gene B and would prevent this enhancer from activating gene A, even though they're actually as far away from one another, but they're separated. And so if you have a CNV that removes this boundary, well, what can happen? Our hypothesis was that this would then allow this enhancer to inappropriately activate this gene. And so ectopic gene expression, a few years ago, somebody was looking at this in another context, not with these domains, and they called this enhancer adoption. More recently, so you always have to have sexy titles to publish in good journals, and some group said enhancer hijacking. I thought the next thing was probably enhancer terrorism, but no. And anyway, so we looked at this, and in fact, the CNV that we found was right on one of these domain boundaries. And I don't have a slide for this, but Malte went on, he got some cells from some of the patients, and he showed that the expression of the PDX1 gene was increased by a factor of about 100. And so, anyway, so that Malte then came to me, and we started to talk and said, well, can we see how common this is, and with using bioinformatics? And so here are the 2 theories that we'll be testing. Haploinsufficiency, gene dosage, I told you about this. So our CNV patient has HPO phenotypic features 1, 2, 3, 4. This gene, so if you have a nonsense mutation here, you get HP3. If you have a nonsense mutation here, you get HP1 and 4. The other theory is topological domain boundary disruption. And here we have a tissue-specific enhancer. And here we have genes that have phenotypes in the same organ that this enhancer is specific for. And so we started to look. So the DECIPHER database is at the Sanger. They have about 20,000 CNVs in all. Approximately 2,300 of these cases are a deletion and are annotated to HPO. So the database is using HPO for phenotyping. Looking at these topological domains, there's about 1 per megabase. So our first thing was, are CNVs enriched in these domains? Well, actually, that we were kind of discouraged because there are about 3.6 megabases on average. So it's not that surprising that they do hit some of these boundaries. And in fact, it was 3.3. But this wasn't different from when we just randomized. So we said, oh no. But then we got some data from the Wellcome Trust Case Control Consortium. So these are about, I think, 5,000 individuals with adult onset diseases like asthma. And so they don't have congenital malformations. And here we just said, this was the observed number of these deletions that hit the topological domain boundary. And then we took CNVs of the same size and did lots of randomizations. And this is what we saw. So it's a pretty big difference. And for the DECIPHER data, actually, there were somewhat more boundaries than you would expect by chance. So this was kind of encouraging. But we also saw evidence of gene dosage in that actually there were less genes contained in these sort of normal CNVs than you would expect by chance. So we had evidence for both things. And after thinking about this for a while, we said, well, in Liebenberg syndrome, there was a tissue-specific enhancer for arms and legs. So it's active in the arms where we see the phenotype. And the gene Phenix1 has something to do with the phenotype. It has something to do with the skeleton. And so we said, that's a signature that we can probably get bioinformatically. And so to do this, we took DNase 1 hypersensitivity data. So basically, if you throw in DNA and this enzyme, the enzyme will cut in places where the chromatin is open. This usually indicates that it's active. And so we saw 2 types, and this was the work of Mike Love, who was at the Max Planck in Berlin. He's now in Harvard. And Mike saw 2 types of peaks, one being a ubiquitous peak. So this is basically the promoter of this gene, and the colors represent these tissue types. So in all tissue types, we see this peak here. Interestingly, these are enriched for, the black color means promoter. So these are the ubiquitous DNase hypersensitivity sites, and we have a statistical method that Mike developed that basically finds peaks that are specific to just one tissue. And so this blue is fetal brain, and so this peak is pretty specific for fetal brain. There's not too much in the other tissues. And looking at these peaks, so here you see the various tissues, they hardly exist at all at promoters, which in general provide basal expression, but they're highly enriched in introns and intergenic regions, which are where you expect enhancers to be. So it's great, we have tissue-specific enhancers. And to make a long story short, we were able to get a list of 20,000 enhancers each that were specific for 10 different tissues. We then said, let's match the enhancers in these tissues to the corresponding abnormality of this tissue, so there's an HPO term. We can then match this with all of the descendant terms. So for instance, abnormality of the heart, well, ventricular septal defect is a descendant term of this, so we can sort of match this to the tissue, right? That's what ontologies are good at. And so we then assign deletions to the category topological domain boundary disruption, TDBD, if 1, they overlap a boundary, 2, a phenotypically relevant gene was located to the left or the right of the deletion. So phenotypically relevant gene means a Mendelian disease of that gene affects the same tissue, right? So skeleton, Wiedeberg syndrome, and a tissue-specific enhancer is located on the other side of this deletion. Okay, and if so, we assigned it to TDBD. The alternative hypothesis was gene dosage, and here we look for genes that phenotypically relevant, so Mendelian disease associated with that gene affects the same tissue, located within the CMV. And we just said, how many do we find? And amongst these 900 cases, essentially using about 6 or 7% of all CMVs are better explained by the TDBD than they are by gene dosage effect. We of course did a lot of simulations, and so the blue is what we observed, and here is when we permuted the phenotypes of the CMV. Here's where we permuted the phenotypes of the individual Mendelian disorders. We did a bunch of other simulations that I'm not showing, where we basically randomly placed CMVs and so on. It was all significant. I'm not going to show you the detail, but we also are able to map mouse and fish data to human data, and that gets us just more genes. So for instance, there are about 5,000 genes with mouse knockouts that have phenotypes for which no human Mendelian disease is known. So we can explain more things, and then up to about 12% of all CMVs can be explained by TDBD. So we think this is actually pretty important. To date, medical interpretation of CMVs is basically just looking at gene dosage, and people will look at all sorts of databases. But we think that it might be important to actually also think about this enhancer hijacking effect and TDBD disruption. So, okay, some examples. So we found this basically automatically with our software. Here are 3 deletions in DECIPHER. They basically take out this topological domain boundary. Here's a gene called FOXG1, and if you have a missense mutation here, you get a variant of Rett syndrome, so severe mental retardation. And these patients had a very similar phenotype to that. And actually, here is a brain enhancer, and so that's a potential expression. And actually, some other groups showed that in cell lines derived from patients with these deletions, they actually get an upregulation of FOXG1. And so we think that, and they said this is some sort of, there must be some silencer within the deletions. We think that this enhancer adoption is a much better hypothesis. Here's another, perhaps surprising, to me it was surprising. So here's a deletion, lots of genes, we couldn't figure out what they would have to do with the disease. The patient had kidney cysts. And we saw here kidney enhancers, and here we saw FGFR2. And we said, oh, that doesn't really sound very renal. There's a bunch of skeletal diseases, but actually the FGFs and FGF receptors are expressed in the developing kidney, and in mice experiment or rat experiments, if you overexpress them, you can induce renal cysts in these mice. And in fact, there's some very rare genetic syndromes with mutations in this gene that occasionally get cysts. So okay, so that's the end of that topic. I hope I convinced you that incorporating phenotype into genomic analysis can help you to address some questions. My lab also does exome sequencing for disease gene discovery, but more and more for diagnostics. And we were asking, can we use the APO for this purpose? I should say, the reason I'm here is actually I met Aaron and Jeff, and they independently came up with an idea which is essentially the same thing. And Aaron, so thank you. And but this might be a slightly different angle. And anyway, so I guess I don't have to tell you much about this, but diagnostics in genetics is still very difficult. And when it talks about a diagnostic odyssey, and the typical diagnostic procedure might involve chromosomes or array CGH, targeted Sanger sequencing. If you do all of this and just throw the book at the patients, you might get up to a diagnostic rate of about 50%. And So some groups have been starting to use exome sequencing in a clinical context, and I think the paper that has attracted the most attention was this one from the Baylor group. So the first author was Yang, and they looked at 250 programs who had been referred for exome sequencing, and their success rate on all was 25%. I no means to be critical of this, but I think a lot of the patients that were recruited in this study had diseases that we would not do exome sequencing on in Berlin. So Ehlers-Danlos, epidermolysis. I mean, if you're a geneticist and you don't recognize epidermolysis bullosa, well, go back to medical school. And I suspect that a lot of this was not, we have no idea what this is, but it's easier to do exome sequencing than a bunch of Sangers. So we have a diagnostic rate on I think, a much more difficult patient group that's 3% better, but I think actually it's a much better result. So I'm sorry if anybody's here from Baylor. Phenix is our program, and so Phenotypic Interpretation of Exomes, and it's a little bit more. So the project also involves some wet lab things, but essentially, well, I got a wonderful email this morning, so we were accepted by Science Translational Medicine, so now the website will be public as soon as I get back to Berlin, and but you enter a list of HPO terms here, and you upload your BCF file, which could be an exome or it could be the panel that I'll introduce, and if you want, you can filter on mode of inheritance and frequency, and you hit submit, and you get you're going to get a ranked list, and so we did this a little bit differently than the Baylor group did. So we said, you know, I mean, in our experience in Germany, you know, if a mother comes in and her child is sick, and you do some tests, and you tell her, well, we found a mutation in a gene called, you know, mothers of pentaplegic. It's a candidate gene in zebrafish, and maybe it's the cause of your child's disease. Well, you would probably get sued, and it's not always useful to mix the research world and the diagnostic world. So this is really just for diagnostics. And we said, well, actually, only about 3,000 of our protein coding genes are interesting in that context. So roughly speaking, 3,000 protein coding genes have been associated with one or more Mendelian disease. And in theory, if we have a pathogenic mutation in any one of these genes, we can go to the HPO database or OrphaNet or OMIM. We can read about that disease and that gene. We can say, does this actually match my clinical intuition? And so, so we basically, with Agilent, designed a panel. We're calling this the Disease-Associated Genome, or DAG, version 1 has 2,742 disease genes. We've developed a version 2 which has a bit more, but so this is a clinically relevant mini exome, and we've been sequencing 6 of these samples per high-seq lane, and we're getting a coverage before we move to the throughput of about 360. And you know, the problem with exome sequencing, if you get a coverage of 50, well, that sounds pretty good, but you're going to have a lot of regions with a coverage of 500 and a lot with a coverage of 2. And you need typically 20 or maybe even 30 reads to confidently call variants. And using this panel, 98% of the target region was covered by at least 20 reads. So here's a panel, and so technically speaking, we think this has a lot of advantages over a typical exome. It's cheaper, and it gets you the stuff that you can actually interpret better. Okay? And so anyway, to upload, you can upload a BCF file resulting from this panel or from a whole exome, but the software is just going to evaluate the genes with known, so it's not going to find you any new disease genes. You get something that looks like this. So we found actually that if we ever do make a diagnosis, it's within the top 20. Okay, I'll explain that in a bit, but so you'll see a web page. It'll have 20 candidate genes. The software first translates from chromosomal coordinates, so VCF file into transcripts. So we just published something called Janovar, which is, we think, much better than Anovar that a lot of people are using. And so this is a missense mutation here. We looked it up, if possible, in HTV or ClinVar, and a lot of mutations are not in these databases, and so we're using pre-calculated numbers for mutation taster, polyphen-SIFT, and we're evaluating the frequency of the variant. So if it's common, it's probably not the cause of a rare disease. And then I showed you at the beginning the phenomizer, so we're basically calculating the phenomizer score. And we're also showing all of the diseases in OMIM and ORPHANET that are associated with this gene. And the gene is finally ranked by, well, in this slide, it's the average of the gene relevance score, which is the phenotype score, and the variance score, which is formed by the pathogenicity and the frequency. And we're actually also saying, well, you know, if this is great, but I just had one heterozygous mutation, and this disease here is recessive, well, downweight this. Right? And so there's a couple of bells and whistles, but I'll skip that for now. So how do we do? Well, we simulated this by essentially, we took 50 VCF files that I'll show you in a second from patients with known diseases. So this was a technical validation, and we removed the known mutation, and then we went to the HGMD, the Human Gene Mutation Database. It has about 100,000 mutations, about 30,000 of which are missense. And so you can get the mutation and you can get the disease, and then we went to the HPO database and took the HPO terms for that disease, and we could then simulate Phenix, right? So then we asked, if things go really well, the gene that we spiked in will be in first place, right? And so that's top, the blue guy here. If things go pretty well, it'll be in the top 5, top 10, or top 20. So these are the three colors. And what you see here, in this evaluation, we looked at just looking at the variant. So we ranked the genes by just looking at the variant. And actually, this performs pretty lousy for exome. So the right gene was, maybe 3%. And even if we're looking at the top 20, the right gene was only in about 35% of cases. If we use the phenomizer evaluation, we get pretty good boost, and the right gene is in first place in about 80% of cases. If we take the combination, it gets up to about 90%. This is actually unfair because in our simulations we were using all terms, and some diseases have about 100 annotations in the HPO, and so nobody's going to do that. So we limited here to 5 terms, but I think this is more realistic. We limited the number of terms to 5, and we took 2 of these terms and made them less specific. So instead of having, let's say, arachnodactyly, we would have abnormality of the finger. So this simulates the case where the doctor doesn't really know what something's called, but he knows sort of where it is. And we also added noise, so this hiccum dictum, just an unrelated term or 2. And still, and here, just using phenotype was a little bit worse, but still the combination was actually still pretty powerful. And so actually the correct gene was ranked in first place in about 86% of cases. To be honest, I think that this, it's hard to do simulations like this. I think this is probably a little bit better than what you would expect. But here's our technical validation. These are cases that we saw over the last couple of years when we made a diagnosis with Sanger. Some of these are easy, to be honest. So here's kind of one, a couple of other things that you don't need to, but this was technical validation, and I would suggest that this is similar, well, I don't know, this is similar to some of the diagnoses that they made in Baylor. So here's Kabuki, no, that's not Kabuki, that's, oh God, I've forgotten my list of 20,000 genes. Whatever. But the average rank for the known cases was about 2.5 or so, and we then said, let's get some hard cases. So we have a really, really good clinical geneticist in East Horn, and so I'm just always amazed at how good she is in making diagnoses and understanding things. But as I say, she doesn't make the diagnosis all the time, and So these are patients where we didn't even have a name. We didn't say we think this might be Noonan syndrome, or we think that, but we had various more or less complex phenotypes without a name. And these kids had been seen, some of them 5 or 6 times with various types of diagnostics, but no answer. And anyway, so we developed sort of a clinical workflow. And again, this is not intended to be a research tool, it's intended to be used. And so for clinics, and so up here, suspected genetic disease, that means somebody comes into the clinic, we say, well, this is hard, I better do the phenotyping, record the phenotypic features, do sequencing, and this here would be the algorithm I mentioned. This gives you a list of 20 top-ranked candidates. And then we soon found that it paid to have a genetics counselor or another genetics professional look at this list of 20 candidates and stop, actually, at the 20th. And in many cases, you would click out to OMIM or read a paper. But if you find something that you think is good, then validate it by Sanger. Look for co-segregation in the family, and if so, make a diagnosis. We write a letter, and that's what we consider success, and that's what I'll show you in a second. If this fails, so it actually never failed, we think that NGS is actually better than Sanger right now, but if co-segregation fails, in a few cases we go back again. And this is what we got. So 28% of these 40 cases, so 11 were solved, and we actually didn't know most of these diseases. So we didn't know what Wiedemann-Steiner syndrome is. Well, okay, we know here mental retardation, so we know what that is, but we had no experience with the type 41. Anyway, so you can make your own judgment as to how difficult these diagnoses were, but essentially that's the results that we got, and subsequently we've done about another 90 cases, and we're still getting somewhere around 30%. So that's what we, and for the difficult cases, I suspect that if we also took these heterogeneous disorders, we would be getting something like some number. So if you're considering using the HPO for your work, why might you want to do that? Well, a lot of groups are now using this at the Sanger Institute. DECIPHER, DDD are big exome databases, CNV databases. The NIH Undiagnosed Diseases program has been using the HPO and also the Exomizer. 100,000 Genomes program just found out that UMLS, so the indexing system for NCBI, will be using HPO. So it's become rather a very international project with a lot of collaborators, and we welcome collaboration. We had, just as an example, we had an Uptake paper in nucleic acids research with 50 co-authors, so a lot of people are interested. If you're going to be doing a study where you're going to be capturing phenotype, this software from the Budno group, Phenotypes, is really cool. You might want to take a look at that. Another thing I'd like to point out, sorry to abuse your patience, but we're part of the Monarch Initiative. This is an NIH project that is looking at model organisms, and so we basically are making semantic annotations for phenotypes of humans. That's what we're doing. Mouse, fish, fly, worm, and basically trying to combine this into one integrated universe. I'm not sure if I have a slide here, but yeah, we've used this to, so I presented today Phenix for clinical diagnostics, but we've used this to make a program called the Exomizer that's looking for novel disease genes. And in fact, humans have 3,000 disease genes that are known, but if you look to model organisms, there's about another 10,000 with a phenotype. And so if we can go back and forth, we've been actually, a few people have used this to apparently help them find new genes already. So if you're interested, here's a plug by the book. And a lot of people have contributed to this work, more than are listed here, but the people who did most of the work for Phenix were Thomas Schemoite. The people, and Sebastian has been drilling through all of these things. Jonas did the work on the CMD project. And anyway, so thanks for your attention.